Here is the breakdown of cellular respiration. The first step is glycolysis, where glucose is a reactant. The products created by glycolysis are 2 pyruvates or pyruvic acids and 2 ATP. The pyruvate from glycolysis goes through the outer mitochondrial membrane in the organelle and reacts with oxygen to break down further to release more energy. The pyruvate is oxidized to form carbon dioxide and acetyl group. The acetyl group and coenzyme A creates acetyl-CoA. Acetyl-CoA starts the process of the Krebs cycle and is oxidized for ATP synthesis. The Krebs cycle consists of 2 acetyl-CoA, which produces 2 ATP and 6 NADH and 2 FADH2 and 4 carbon dioxides. Glycolysis in the Krebs cycle produces roughly 4 ATP molecules per glucose molecule through the process of substrate level phosphorylation. Substrate level phosphorylation can occur in the cytosol and mitochondrial matrix and is less efficient than oxidative phosphorylation. Oxidative phosphorylation occurs in the inner mitochondrial membrane and is responsible for making nearly 26 to 28 ATP, which is a sufficient amount of energy.
Go.
Finally, I will now expatiate on my oxidative phosphorylation model. The electron transport chain consists of complex 1 through 4. 1, 2, 3, 4, the electron transport chain. Whereas the ATP synthase is a part of chemiosmosis. Both combined make up oxidative phosphorylation. Without one, there can't be oxidative phosphorylation. Furthermore, complex 1 only focuses on oxidizing NADH to NAD+, while losing an electron. The hydrogens will move towards the inner membrane space while creating a proton gradient, as we can see here. NADH in complex 1 has a higher energy level than FADH2 in complex 2. Complex 2 strictly focuses on oxidation of FADH2 to FAD, which indicates losing an electron. The excess NAD and FAD will be recycled back in the process of glycolysis and so on. The electron transport chain will carry the electrons through the complexes while passing through cytochrome Q and site C. As you can see here, there's an electron. The last cytochrome of the chain donates its electrons to oxygen as it's quite electronegative in complex 4. Each oxygen atom also picks up a pair of protons, which neutralize and buffers out the -2 charge of the added electrons, which in turn produces water in the electron transport chain. So we see water is produced. With all of these lovely protons in the inner membrane space, potential energy is formed and creates an environment for facilitated diffusion to help bring hydrogens down the concentration gradient of ATP synthase. While this process occurs, ADP and inorganic phosphate create lots of ATP. For an analogy and a creative standpoint, I would like to share 4 trees as the 4 complexes in the electron transport chain. So here are 4 trees. Trees will lose oxygen, and us as consumers will gain paper. When paper is gained, more books can be created. Woohoo! Finally, I did not use the term Christe in this oxidative phosphorylation model. However, it is very important to the electron transport chain as it creates more surface area